All right, so section 5.6 talks about types of energy. And I kind of already, I can't say I kind of already because I did this in reverse order, but let's talk about the two types. So we have kinetic energy, which is energy in motion, and we have potential energy, which is stored energy. Chemical energy, when we talk about chemical energy in a biological context, we're generally talking about the energy between bonds. So we're talking about those covalent bonds in our biological macromolecules, those carbon-carbon bonds in our lipids, the carbon bonds, the carbon hydrogen bonds in our sugars, all of those covalent bonds that we find in sugars, proteins, and lipids that are used for providing us energy, fuel to make ATP. And that ATP is going to do the kinetic energy, it's going to do the work. So free energy is how much energy can we actually use, because we cannot transfer all of our energy, all the energy that we transfer. In the next section, you'll learn about the first law of thermodynamics. We can't transfer all of it. So the part that we can, we call usable energy. We have two different types of chemical reactions. We have what are called exergonic or endergonic. Exergonic reactions give off, they release free energy. That means energy that we can use to do work. Endergonic means that these are chemical reactions where I am not going to happen unless you put work into me. So they use free reactions. So lots of times we do what's called coupled reactions, where we will use the energy release from one reaction to do the endergonic. So the exergonic reactions are used to complete or be able to do the endergonic reactions. So we can see here, as we break down those molecules, we're releasing that energy, and then we can use that energy to put the molecule, because catabolism usually is an exergonic reaction. It usually releases energy as you're breaking those bonds, and versus anabolism is generally an endergonic reaction. Because again, we're going to be opposing entropy, which I will talk about in the next lesson. So ATP and living systems. So ATP in living systems is adenosine triphosphate, has 3 phosphate groups. So it has our ribose group, our adenine, nitrogen base, and then 1, 2, 3 phosphates. And so these phosphate groups, they do not like being by each other. It takes a lot of energy to force those bonds into place. And so by relieving those stress in those bonds, by breaking those bonds, we can release energy. This is an exergonic reaction. Now, to put it back together, so this would be like doing work on our cell phone, and then we're done with work, we're empty. Now we got to charge it. And so that's an endergonic reaction. And we're going to recycle our ATP between ATP and this is ADP, because we have 1, 2 phosphate groups. And so this is going to be like our battery, but these are chargeable batteries. So we're going to charge them, use them to do work. And then once they're out, we're going to recharge them again. So as I mentioned, we take our foods in, we can use our proteins, and as we break those down into their monomers, amino acids, right? We can then, that would be catabolism, and we can then use that to do work. We do not want to do cannibalism, which means we're breaking down our own proteins because we're not getting enough calories. Right. So if we need to, we can start to break down our own proteins if we are starving. Right. And then we also have this process where we're breaking down, again, catabolism, we're breaking down sugar. We can also store sugar and glycogen. So we can kind of go around and around on that one with the liver. And we also have our fats, which we can break down, or we can store. All of these are going to be used to make ATP. So when we talk about ATP, its structure and function, and we're going to learn exactly how we make ATP in the next chapters. But what we're going to do is there is a protein called ATP synthase, and we are going to have that do work, and I don't want to give you too much detail here, but basically this is the job of that enzyme, and that job of that enzyme is to put that third phosphate onto ADP to make ATP. So that's to charge our battery. This would be an endergonic reaction. So we will need something to fuel this, to run this in order to make ATP. And we're going to learn how we do that. ATP hydrolysis, that would be our exergonic reaction. And so we saw this with our sodium potassium pump. As we put our ATP on there, we're going to break off that phosphate group, and that's going to release energy. And that energy is then going to be used to drive the sodium against its concentration gradient in active transport. So how living things use energy? Well, again, we couple them. So we're going to use those exergonic reactions to power the endergonic reactions. So we're going to store energy. We'll have that ATP that's going to be available to run that sodium potassium pump. And then the last section in this presentation will be on enzymes, and that is what you're going to be doing your catalase lab with your bubbles. And so with that, you are going to be using an enzyme. Enzyme is most of the time a protein. Sometimes it can be RNA, but most of the times, like 97%, it's proteins. And what they're going to do is they're going to speed up that activation energy. I kind of already mentioned this before, but now we can see this little table here. So this is an exergonic reaction. It's exergonic because the reactants are higher, and when we get done, our products are at a lower energy state. So the amount of energy we give off, our free energy, is right here. But in order for me to give off this free energy, I got to get up and over this hill. This hill is huge. And so I have to put in energy to get up over the hill to get out this energy here. So an enzyme, what an enzyme does is it lowers that hill so I can profit more, and this will move faster. Okay. So enzymes lower the activation energy. And enzymes are very specific to what they bind to in the job that they do. So here is that picture again. So here would be the opposite. Notice my reactants are lower than my products. So this is going uphill. So this is an endergonic reaction. Even endergonic reactions are going to have that activation hill. So we have the red dotted one is with the enzyme. And then we have the purple one here is without the enzymes. So enzymes, again, they lower the activation energy so that we can get this process going faster so it can do its job in the amount of time needed to support life. Enzymes, like I said, are very specific. They have this shape and that binding pocket. And with that binding pocket, that specific shape that the enzyme has will fit in their substrates. And in this case, it's actually going to put this product together. And so where it binds is called the active site. And so our two substrates bind into that active site. We're going to then force that covalent bond. And now we have, and this would be an anabolic process. Most of the time, we always talk about a catabolic. We have it going the other way where we take a product and break it down. But in this case, Liz wanted to show the opposite because we rarely show this, but it does happen. So we're going to put this together. So here we're going to have the opposite. So we're actually have our active site. We're going to bind to it and we're going to break it into its products, which is usually what we show when this happens. And usually what happens when that substrate binds, the enzyme changes shape, and that conformation change allows this process to happen faster, whether it's bending, putting stress on that covalent bond to break, or holding those molecules together so that covalent bond can form. So that enzyme is very, very specific to what it binds to in that active site. Now we can control enzymes in a couple different ways. And the two main ways we're going to talk about is this thing called a competitive inhibitor and a non-competitive inhibitor. And I really like pictures much better than words. So here's our normal enzyme activity. Notice that our competitive inhibitor is going to slow it down, and our non-competitive inhibitor even slows it down even further here. So what do we mean by competitive and non-competitive? So a non-competitive inhibitor means it does not compete for the active site. Notice this is the substrate and it cannot bind because a second molecule is blocking it. It's sitting in that active pocket saying, nope. We can say not in my house, right? Anyways, I'm getting silly. So after making 6 of these videos. So we have this competitive inhibitor here. And so the substrate can't bind. And so we can't do the job that it was supposed to do. In this case, there is a second binding pocket on this enzyme, and that is often referred to as allosteric control 2, and allosteric refers to changing its shape. And so what happens is when we bind to this pocket, it's not competing directly for the binding site, but it changes this pocket in a way that, so this pocket changes shape. And now my substrate does not fit. And so it's saying, nope, you cannot bind. And so these are two ways that we can control enzyme activity. We can either compete directly or we can compete indirectly in our non-competitive inhibition, but they're both inhibiting, slowing down the rate at which this enzyme is working. So we also, when we talk about non-competitive inhibition, so an inhibitor, like I said, it binds to that alternate state. Like I said, we also refer to this as allosteric because it changes that binding pocket. It changes that shape to that enzyme. Now we can see where we can inhibit, where we change the pocket, or the pocket has to be changed to be activated. So we have allosteric inhibitors and we have allosteric activators. So there are all these little different ways to control these enzymes. We also have what are called cofactors and coenzymes. So many enzymes need a little bit of help to work at their optimal speed. And so they have these other molecules that bind to them to help them to fold, whether they help them fold in their best possible shape, their best conformation. Conformation is another word for shape. If it's a cofactor, it's generally inorganic, and inorganic ions are generally your minerals. So sometimes that would be like copper or zinc, or we talk a lot about zinc, right? Zinc is a big cofactor for your immune system. So these would be iron would be in hemoglobin. Coenzymes, these are organic molecules and these are your vitamins. And so here's some of your dietary vitamins that are very important. A lot of these are B vitamins that help again with your immune system, and they help with those enzymes work properly, or other types of molecules work properly. And the last thing we're going to talk about here is feedback inhibition. So feedback inhibition is another way of controlling. And I always think of like an assembly line. You can't have your assembly line going too fast because then people aren't going to be able to complete their job quick enough and it won't get done by the time it's to the next person. Or you also don't want, that person standing around waiting. That's not being very productive. So we have ways to regulate this. And so if we start here with our inhibition, so we're going to start with our substrate here, which is our green triangle. It binds to our enzyme, and our enzyme is going to change it to this purple rectangle. The purple rectangle binds to another enzyme and it gets its shape changed to this blue, I don't know what shape you would want to call it. And then that binds to the next enzyme and that changes it to the square. And the square may say, you know what, we have enough yellow squares, and so we don't need to continue this process. And so this is called feedback inhibition. This is like your thermostat at your house, right? You set your house at your temperature, your house gets cold, the heater kicks on, you have enough heat, it turns it off. And that's what it's saying. Okay, we're on, we're all go, 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 go. Oh, wait, we're good. We don't need any more. And so this is called feedback inhibition. And like I said, that was the last thing we were going to talk about. And so again, this presentation was, the main focus was enzymes. We will be doing a couple enzyme labs to help with understanding this process. This chapter do notice this chapter does have 2 labs at the beginning of the module, the home module for chapter 5, because we are doing 2 labs that are a part of these concepts in this chapter, because there's 2 main big concepts, and that was osmosis and diffusion and then enzymes. All right. So I hope this was helpful, and I will see you in the next lesson. Bye.